Hi everyone, thank you for this opportunity. My name is Marie-Elyse Lafaille-Magnan and I'm going to share with you how mothers' prenatal and postnatal depression symptoms act in consort with genetic risks to predict ADHD symptoms in 4 to 6 years old. Imagine pregnancy disrupts your life. How do you feel? Are you tired, lonely, depressed? What if the feeling never went away? Could there be a relationship between the exposure to mother's mood and ADHD? Could there be an interaction between the exposure to mother's mood and genetics? Attention deficit hyperactivity disorder is hereditary. The number one biological suspect is the dopaminergic pathway. There's a large range of prevalence. There are no standard scales used. A child's function is usually evaluated in different settings with multiple scales. For research, this is burdensome because studies are rarely comparable. Latent factors provide a method to reduce data dimensionality from multiple scales, raters, and time points. We combine multiple assessments of ADHD into one measure. We use data from 394 mother-child dyads from the MAVAN cohort conducted in Montreal and Hamilton. We created a latent factor for ADHD with parent, teacher, child ratings, and diagnostic measures. We tested the constructability by examining the ADHD factors association with comorbidities, sociodemographic, and risk factors. We obtain prenatal and repeated postnatal ratings of maternal depression. We use the area under the curve for mothers' experience of postnatal depression over time to collapse repeated measures over the first 6 years of their child's development. Mothers had greater symptoms of maternal depression in children with a high ADHD factor. We used the latent environmental and genetic interaction package to predict ADHD, separately for the polygenic risk score for ADHD and dopaminergic variants of children, while adjusting for maternal education, maternal age, sex of child, study site, and the first three principal components from genetic analysis, which explain ancestry. We found that younger and less educated mothers had children with higher ADHD factor scores. Higher ADHD factor scores were associated with greater sleep impairment and psychiatric comorbidities. It appeared that in those exposed to less prenatal depression and greater postnatal depression have higher ADHD factors in boys, but not girls. In models exploring the effects of the polygenic risk score and maternal mood, we found an interaction between sex and maternal depression, but no effect of the PRS. In models where we investigated the three-way interaction between gene, environment, and sex, again, we only found a sex by maternal depression interaction. Meanwhile, when we looked at models that investigated dopaminergic variants, we found an effect of the genetic susceptibility and an interaction between the genetic susceptibility and maternal depression. When we looked at the interaction between dopaminergic variants, maternal depression, and sex, we observed a significant gene by maternal depression interaction. What was interesting is when we loaded a prenatal by postnatal depression interaction in the environmental latent factor, the gene by environment interaction disappeared, but we gained a significant gene by sex and gene by maternal depression and sex interaction. Our main takeaways are that we can use factor scores and the area under the curve to reduce information, which makes it easier to compare results with other studies in the future. Sex is an important factor to consider in studies of mental health. Prenatal and postnatal maternal depression both matter. Congruence of prenatal and postnatal depression appear to be informative. And dopaminergic variants are more robust predictors of ADHD than the PRS for ADHD. This work can inform guidelines for supporting mothers before and during the early childhood development. Finally, thank you to the MAVAN participants and group and collaborators. Thank you to Ashley Wazana's lab. And thank you to all the funding for this study.